Hello, we're glad you've joined us for this live webinar, Accelerating Gene Editing Research Using the Power of Flow Cytometry. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit www.thermofisher.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Natasha Jacobsen, the Product Specialist of Flow Cytometry Instruments at Thermo Fisher Scientific. Welcome, Natasha. You may now begin your presentation.
Hi, thank you so much for joining. So today I'm going to discuss next level CRISPR gene editing with flow cytometry and how you can use the power of flow cytometry to streamline and enhance your CRISPR gene editing workflow. So a little more specifically of what we will cover today, we'll discuss some of the traditional methods for analysis of gene editing experiments, and then we'll talk about how flow cytometry can be used for rapid high throughput analysis of these gene editing experiments. We'll also discuss some of the unique benefits of using flow cytometry in the gene editing and cell engineering workflow, and then also highlight and touch on some of the additional applications in the genome editing workflow, such as viral titering, viral transduction optimization, and fluorescent gene tagging. All right, so let's start with discussing what gene editing is broadly and how it's done. So gene editing is the process whereby you add, remove, or replace a piece of DNA. And how is this done? So the most widely used method and most efficient method currently is using the CRISPR-Cas9 system. So the CRISPR-Cas9 system is an RNA-guided DNA nuclease system that has two major components to consider. So one is the Cas9 protein, and this is the protein that actually has the nuclease specificity, the nucleus activity that allows you to create a double-stranded break. And the CRISPR RNA is the target-specific portion. This is how you actually target exactly where you want your precise edit to occur. And this happens through Watson-Crick standard base pairing rules. So as you can see, you have the target-specific CRISPR RNA that's complexed with the Cas9 to target a specific portion of the genomic loci. There's really one major thing you need in order to target a specific portion of the genome, and that's a PAM. So the PAM is an NGG, so it can be any base followed by two Gs, and this is required for Cas9 to bind. And any portion of the genome that has this NGG is now targetable by CRISPR. The standard CRISPR length is 20 nucleotides, but that can vary. But that is the most commonly used CRISPR length. And then there's also a tracer RNA, and this combines with the Cas9, and we generally create a fusion, and we collectively refer to this as a guide RNA, which is the target-specific CRISPR RNA with the tracer RNA. And these are the two major components that are required to target anywhere on the genome that has a PAM. So why do we care about this? I mean, really the opportunities for research and, even cell and gene therapy using CRISPR they're really endless. You can use this to create animal disease models, tissue and disease modeling in vitro, This can also be used for stem cell engineering, gene therapy applications, and even for agricultural studies to create, disease-resistant transgenic plants. So let's get a little more into the molecular pathway of gene modification and, how this occurs on the molecular level. So you begin with your piece of double-stranded chromosomal DNA. And from here, when you add your Cas9 and guide RNA complex, you create a gene-specific double-stranded break. And at this point, there are two pathways that the cell can go through. It's the cellular endogenous machinery that repairs this double-stranded break. And one is non-homologous end joining, or NHEJ, in which the cleavage is now, the endogenous cellular machinery takes over, and it either adds or deletes portions of DNA, creating an indel that refers to insertion or deletion. And this creates a knockout. That's the overall goal. And that's really the standard CRISPR workflow. When you're just adding a guide RNA and Cas9, your goal is generally to create a knockout. And one thing that's important to note here is this is not a controlled process, right? This is random within the cell, how many bases get added or deleted at this time. The other pathway, which is a little more complicated, is homology-directed repair. And this is where you can create precise edits. And this is done very much in the same way. You still are using a guide RNA and the Cas9 protein. But in addition, you also add a donor template. And this allows for a precise repair based after your CRISPR modification. And you can see through this little drawing here, you just require, correctly designed donor template with appropriately designed homology arms, and then you can end up with a precise modification. So this is very advantageous for if you want to, let's say, mutate a promoter or add a promoter, or even endogenously tag a gene or add a gene. And, one thing that's, noteworthy here is homology-directed repair. It happens generally in a much less efficiency, a lower percentage. It's a little harder to get a large number of cells that have this precise modification. And that comes into play a little bit later on some of the things we're going to talk about. So let's, take a, little more of a deep dive and look at the gene editing and cell engineering workflow. So there are a lot of different reasons to do gene editing and a lot of different, experimental endpoints, but most gene editing experiments will follow this basic workflow, which is to design, deliver, detect, expand your clones, and validate. So during the design process, you design your guide RNAs, order, purify, or synthesize these guide RNAs. And then the next step would be to deliver. So this would be to prepare your cells, optimize your transfection efficiency. And we have a variety of different tools available to do that, whether you're using neon transfection, if you're using the our Cas9 protein, lipofectamine CRISPR-Max. There are a lot of different options, including, viral delivery. That's, another option for delivery as well. From here, you need to detect your editing efficiency of your edited pool. So, if you recall from the previous slide, I mentioned that the mutations that are created using CRISPR-Cas9 are random. So you need to analyze the editing efficiency. Not all CRISPRs are created equal, right? And confirm the most appropriate guide RNA to be used for your experiment. From here, you now need to expand your clones. So, depending on your application, quite often the end goal is to get a clonal population of an edited cell, in which case you'll need to, go down to single clones and screen and re-rack these clones. And then the final step, of course, will be to validate, right? to ensure that you have the appropriate edit and the appropriate phenotype of these edited cells. And this is done through genomic validation using NGS sequencing, genomic cleavage detection, Sanger sequencing, as well as off-target analysis and functional validation. And we'll go through all of these in greater detail as well. So, what I'd like to highlight here is this is really, a collection of tools and the standard workflow. And flow cytometry can really play a role and help accelerate this, in every step of the way for each of these major and frankly, laborious steps through the gene editing workflow. So you can use flow cytometry to help validate your guide RNA design. You can use it to help optimize your delivery, as well as quantify your editing efficiency, and expedite the overall screening and clonal isolation process, as well as get that functional validation of your edited clones at the end. And we have a variety of imaging tools and a variety of instrumentation to help with this as well. So I'm going to talk a little bit, or quite a bit rather, about the Attune NxT flow cytometer, a little bit about how that partners with EVO cell imaging in your functional analysis of your edited cells, as well as some of our viability reagents and, of course, our antibodies for functional validation. So let's take a little bit deeper dive into the traditional methods of editing efficiency analysis and this overall workflow. So you can see here the timeline along the top. I have about an average representation here of 21 days, and this is from start to finish to create your validated cell line. Some can take much longer, and there's some cases you can go a little bit quicker, but that's an average. So your first step is to edit your cells by delivering your CRISPR-Cas9 complex to your cell line or your primary cells. From here, you now have NHEJ occurring, and now you have an edited pool, and that's denoted here by the different colors here. So each cell, in theory, can have a different edit at this point, right? You have a pool of edited cells. And then the next step would then be to isolate and expand these edited cells using your, sorting or your limiting dilutions to get your single cell clones. From here, there's, another step of screening, and now you need to consolidate these clones into a 96-well plate with, and be able to, functionally analyze each of these, expanded clones. And then the final step would be to expand, validate, and bank these cells. So through each of these steps, there are a lot of analysis steps, right? So, this 21-day workflow does not only require a lot of time, and a lot of cell culture in the hood, but also a lot of additional wet lab work to, analyze these clones on each step of the way. So I'd like to talk a little bit about some of the traditional methods of editing analysis. So one would be the genomic cleavage detection assay. So this is a really popular gel-based assay that can give you a quick and semi-quantitative answer as far as your gene editing experiment and how efficient it went. So from this step, you go through, lysing your cell pellet. You design primers flanking the region of interest surrounding your edit, and then you go through a denature and reanneal, and then go through an enzyme digestion step that recognizes these indels that we discussed earlier. And then you can run a gel and analyze. And you can see here that this is, just done using band densitometry. And you have a semi-quantitative answer as far as, what portion of your cells are edited based on that uncut versus cut band. So this is, semi-quantitative proportional analysis. So for the next level of accuracy, another option would be to use Sanger sequencing. And Sanger sequencing is also very popular for analyzing your editing efficiency, and it's very quantitative, and it goes through a lot of the same steps. So you go through lysis, PCR as before, flanking your region of interest, But then you have to prepare a sequencing reaction using a big dye PCR and then a cleanup. And then you actually need to run it through the sequencing reaction and then go through analysis. And here I have an example of 2 chromatograms, one of a control chromatogram, uncut, as well as an edited sample. And you can see just before about 210 base pairs, you can see where the cut site had occurred. And you can see that scrambled chromatogram, which is representative of the edit in the indel. And this is run through a program using indel decomposition, where we integrate the area under the curve to give you a nice quantitative estimate of how edited, what proportion of your cells are edited in this pool. The next level of rigor here would be using next-gen sequencing. Very similar, again, as far as the steps. You go through lysis, PCR amplification. It's a little more involved, though, with NGS. You need to, go through a few additional steps, such as adapter ligation, purification, another PCR, which is the NGS PCR reaction and purification, and then prepare the template, and then go through the NGS and analysis. And this, again, is, it's still very useful. It is definitely an appropriate method when you're doing your final quantification and your off-target analysis, but this is a lot of work. All of these steps are a lot of work, and in the meantime, you're growing the cells as this is all happening, taking your lysates out, going into the wet lab, and doing a lot of additional hands-on work and using a lot of additional reagents to do this. So, I'd like to propose and, suggest in many cases, another appropriate method to do this would be to use flow cytometry. So now compare this workflow. If you're already growing your cells in the cell culture, really to analyze your editing efficiency using flow cytometry, you just need a validated antibody, in which case you just stain and wash your cells, and then you can run your samples by flow cytometry and analyze your data. And this is, very high throughput. This is done, using a 96-well plate. It doesn't require any additional reagents except for some, flow cytometry buffer and an antibody. And you get a very nice quantitative, functional knockdown of your gene editing experiment. And so here I've just highlighted, an example of what this data would look like and how you would set up your experimental parameters. So in this case, I'm looking at a histogram overlay plot of a CD47 knockdown experiment. So in this case, I designed a guide RNA to target CD47. I had a validated CD47 antibody conjugated to APC. And you can see on the right here of this histogram, this is an overlay. This is the unedited cells versus the edited cells. And you can see that, shift to the left showing functional protein knockdown. And then I have just a few examples of the plate software, the plate analysis software on the Attune NxT, where you can, very quickly get a nice pictorial layout of the knockdown efficiency. And as you can see, you can test many different parameters and, look at the effect each of these have on your functional knockdown. And on the right, I have an example of, what a gating tree would look like. So the real message here is, you have your protocol completely set up, within your, flow cytometry analysis software within the Attune NxT. And you just run your plate and you have everything you need. Essentially, all your analysis is already done. So, why use flow cytometry for gene editing? So, there's a lot of different advantages for when to use flow cytometry and, when it can really, augment and expedite this workflow. So, one benefit is, it's high throughput, right? So you can use it to, it's a very high throughput method to validate your guides. And, you can collect a, a 96-well plate in about 45 minutes. None of the previous methods mentioned could give you an answer for, a 96-well plate in anywhere near that amount of time. So it's much faster and with much less waste. Another added benefit is it actually gives you the functional knockdown. So all of these other methods previously described are, looking at the genomics, looking at the actual edit on the genome, which, as we know, an edit does not always confer knockdown. So that's very important. Another huge advantage to using flow cytometry is you're getting single cell analysis. So as I mentioned previously, when you edit a pool of cells, all of these cells theoretically can have different edits. When you're using flow cytometry, you're actually looking at a single cell knockdown, and you can use that to know how many of your single cells are actually functionally knocked down. And really the only way to do this using these other methods would be to go to clonal isolation, which, if you recall, is about 3 weeks and a lot of work down the road. Another thing that flow cytometry can give you, information it can give you that these other methods can't, is really guide RNA multiplexing analysis. So you can actually look at, as I mentioned, single cell analysis, but you can look at multiple genes being knocked down in a single cell, right, simultaneously. And you can do this without clonal isolation. Another really important application of flow cytometry is being able to target and look at these gene interactions, being able to analyze these functional gene networks like synthetic lethality and synergistic effects. We know that, these gene networks exist in highly redundant pathways. And, sometimes you may need to knock down a few genes to get the desired phenotype that you require. And, flow cytometry allows you to do that and adds that power to your research. Another very important reason to use flow cytometry in your gene editing experiment is, we're using cell biology, right? We need to be able to look at the viability of our cells, look at the proliferation, see if other effects to the overall cell growth and cell phenotype after edit. So that adds a lot of value as well. So I'm going to go through, one application, and this would be using, gene editing in the PBMC workflow using CRISPR-Cas9 in the Attune NxT. So, the basic workflow here would be to, go through our T-cell isolation and activation. As you see, we have, Dynabeads kits for that. And then go through T-cell editing, the next step. And, and I have a few representative tools and products listed here on the right that can, help this process along the way, like the Neon transfection system, our TrueTag Cas9 protein, as well as our synthetic guide RNAs. From here, you can then do the functional knockout analysis using our eBiosciences antibodies in the Attune NxT. And then you can also use our fluorescent viability dyes to give you more information about your edited cells in the end, right, for the overall cell phenotype. And then, of course, for any validated cell line and in any gene editing workflow, at some point, genomic analysis is important as well, right? And we have all of those tools as well, from our genomic cleavage detection kit to give kind of a quick and straightforward, answer that's semi-quantitative. And then, of course, we also have Sanger sequencing and next-gen sequencing as well. So here I'd just like to, kind of take you through this workflow a little bit and give you an example of, what the data looks like. So up on the top, I have, the workflow stated out again as well. And, you can see that within, it's a 48-hour process to, validate your guide RNA and look at your knockdown efficiency using flow cytometry. So in this case, this is a guide RNA targeting the TCR in PBMCs, and this is conjugated to PE. And as you can see on the bottom left, we have the scatter plot with fluorescence on the x-axis and then scatter on the y-axis. And you can see that the cells that are untransfected, untreated, are 92% TCR positive. And then we have two different guide RNAs targeting the TRAC and TRBC locus that were used using electroporation and Cas9 RMP. And you can see, the statistics right here on the plot. You can see that we have, 97.8% knockdown and 92.5% knockdown, respectively, for each of these. And, this is an answer that is, attainable within, 48 hours of your experiment and, without any additional processing steps. And then, additionally, looking at another piece of data within this experimental workflow is it correlates very, very nicely to editing efficiency by NGS. And, in a lot of ways, it gives a little more information. And these two are used as pairs quite frequently, right? Looking at how efficient you were able to edit the genome versus how efficiently you were able to knock down the functional protein. And you can see that here as well. And, this is really the first step, kind of going back. One of the first steps of any genome editing experiment is to validate your guide RNA designs and pick the best one. And we rarely just design one guide RNA, right? Standard, most people choose about 3, right? And then decide to move forward with the best one from there. So, in the case of using flow cytometry, you can have your answer in 48 hours and then know which is your most appropriate guide to move forward with very, very quickly. So really the next huge advantage to using flow cytometry here, and not just an advantage, but it's unattainable by other means, by standard means, and that's the ability to look at multiplexed editing. So as I mentioned before, in a lot of cases, when you're doing cell engineering and when you're trying to do cell therapy studies, there may be several genes that you need to edit in order to get the desired phenotype and in order to get the desired cell line that you require. And flow cytometry allows you to interrogate these cells and do single cell analysis for looking at multiplex knockdown. So if you remember previously, you have a pool, right? If you're using NGS or Sanger sequencing or genomic cleavage detection, you need to move down to single cells, right? And then you can, be able to know for certain that it has all 3 targets knocked out. In the case of flow cytometry, I can just add all 3 antibodies at the same time, right? That, of course, these are pre-validated antibodies. And I can actually know what proportion of my transfected pool has actually been edited for all 3 targets at the same time. And that is very useful and very powerful. And then in addition to that, another thing worth mentioning is, from the next step here is if I was to do GCD, Sanger, NGS, I now need to design 3 different experiments, 3 different primer sets to amplify each locus, right? So, an N of 1 experiment immediately becomes N of 3, right? And that compounds over time depending on how many edits you want to make. But in the case of flow cytometry, you can see this graph here. This is actually just, one well, N of 3, right? Where I've edited 3 different targets. So B2M, PD1, and the track locus. And I've analyzed them via NGS, GCD, Sanger, flow cytometry, and viability. And you can see that they correlate very, very nicely. The Sanger, the NGS, and the flow cytometry, are very predictive with one another. And then I also got this additional, viability measurement as well. And just remember here, this was N of 3. So with flow cytometry, I just had to put the plate in and run 3 wells. To get the equivalent data here that's represented in this graph via NGS, GCD, and Sanger, I actually had to multiply that by 3 again and look at each of these targets individually, meaning I'm really just getting an estimate here of what I think my triple knockout efficiency would be based on, statistical analysis. But with flow, I actually have an exact answer. I know, approximately 54% of my cells successfully have all 3 of those edits. And that is very, very helpful as well for doing further studies down the line on viability, as well as knowing how many clones I need to plate out to get the cell line that I desire. So another advantage and another level of power that flow cytometry adds is, you can really look at, gene expression over time. So, making an edit to the genome, isn't going to change as much as expression can, right? So we know that, when you're looking at PBMCs, for example, there's a lot of differences between donors. And, again, these cells exist in, these genes are interacting with one another in complicated gene networks. And, expression can definitely change over time. So flow cytometry can give you that additional, piece of information about what the actual expression levels of these proteins are over time. And again, if they recover. So, on the right, I just wanted to give you a snapshot of some of the things that you can encounter in a gene editing experiment. So this is the previous data set, right? 3 different genes, being multiplexed to edit at the same time. And I looked at these both, 3 days post-edit and 7 days post-edit. And this is a heat map set to PD1 expression. So, if you can look at the upper right-hand corner where I've, drawn a box around that portion of the plate. So this is a single knockout. So, 7 days post-editing, my PD1 is still knocked down effectively. However, if you look at in the bottom left of the 7 days post-edit plate, outlined there in red, my PD1 expression recovered over time. And that's really important because when I ran my NGS, when I did my Sanger sequencing, the same proportion of my cells is still knocked out. The gene's still been edited, but it wasn't completely knocked out, right? So now it can still be influenced by other genes that it's interacting with. And, this is a well-documented issue, B2M has been implicated as being an issue for PD1 resistant or PD1 treatment resistant tumors. So, flow cytometry can give you that extra level of power to look at your gene interactions. And, this wasn't something, for example, that, I was anticipating or looking for. And, this was, really important to know that, my expression was not going to exactly correlate with my knockdown because of this gene interaction. So flow cytometry allows us to have a more comprehensive analysis of gene knockdown and, really elucidate these gene interactions that you wouldn't be able to detect easily using other methods. And then, here's just another example of a really straightforward panel. It was a little more straightforward because these genes did not interact with each other in the same way that B2M and PD1 did. And you can see again here, just an example of, the data you get from flow cytometry. You can see the overlays on the top, the unedited versus edited populations on this overlay plot, which is all, already in the Attune software. And then you can look at these bivariate plots and, look at your different populations. And, the flow cytometry also allows a lot of nice visual tools like, hyperlog plots where you can, really look at your expression levels and your populations and help to tease out and find that data. So, so now, moving forward to, another level of power that flow cytometry allows for your gene editing cell engineering experiments, and that is it gives you the option to do viability studies as well. And that's very, very important, right? You're doing gene editing, but in the end, you're looking at your cells, you're looking at your cell health, you're looking at, their ability to proliferate, their viability over time. Additionally, if you're optimizing gene editing experiments, or say, the goal is to create gene editing tools, toxicity is very, very important. So, here's another example of using flow cytometry to get that additional level of data. So here I've used just, two dyes, Cytox Red and calcium AM. One is a membrane permeability dye, so Cytox Red will enter the cells if the membrane has been compromised. And then the calcium AM is a vitality dye. So, you can see that there are, a couple different populations here, right? You have your, the majority of your population are going to be your viable cells up here. They're calcium AM positive and Cytox Red negative. But then you also have, two other populations. You also are going to have some where their, membrane is compromised, but yet they're still, showing vitality. And then you also see the ones that have your, that are Cytox Red positive and calcium AM negative. So those are, truly your dead cells. And, as you can see here, you have a plate map, right off the bat that gives you a visual representation of this viability of your cells. And then, all the necessary statistics as well to look at your cell viability based on different conditions. So it's a really nice way to, optimize your dosage for your gene editing experiments, as well as your transfection conditions, your payload and, your electroporation protocol if applicable. All that stuff is, not arbitrary and can be very time consuming. So it's nice to have this accurate readout. And again, remember, you're still in the same plate. You don't need to create a duplicate plate for another assay. All this data you can get at once. So, and along the same lines, flow cytometry gives you additional data as well. It gives you your count and concentration data, which is really nice. And the Attune is uniquely positioned for this because the Attune NxT flow cytometer is a syringe-driven system. So you can use the Attune to get precise volumetric counts without the need of counting beads, which is, really great. And that not only does it give you that count and concentration data, but if your end goal is to get a clonal population of cells, then at some point you're going to probably go through limiting dilutions, right? And you're going to plate your 0.8 cells per ml or 0.5 cells per ml, whatever your general protocol is, and so you can analyze your data on the Attune flow cytometer and then immediately have a count and, don't need to go through another counting step and then go directly to your plate. And, another really nice thing about having this count data is, for optimizing your transfection, right? Every single method, including neon, right? The zap of the neon is going to kill some of your cells as you go. And, that is definitely, a part of the workflow that is, very time consuming and necessary, right? So, you can see quickly here that, I can, use flow cytometry to also optimize my neon conditions and, see how many cells I'm going to need to start with, in order to move forward because I get that accurate count. So, just along those same lines, flow cytometry can really simplify and expedite this limiting dilution calculation as I talked before. So, your standard process would be to, edit your cells and then, lyse your sample and evaluate via genomic cleavage or sequencing, count your cells and plate by limiting dilutions, right? And, as you can see, I have a big stack of plates there. That is pretty standard for one cell line, depending on the cells that you're using, maybe 10 to 20 plates just to get one cell line, right? And these are, this is media, this is hands, you have to, take care of these over time, space in your incubator, or, you can use the Attune NxT, get an accurate count, even use the Attune NxT sample recovery feature, right? And, use this multi-parameter analysis as well to get immediate and accurate results in one single step. And, what I mean here, that was kind of a lot, is, if I'm trying to get single cell, a clonal population that has 3 knockouts, right? If I use the Attune and I use, you know, my 3 antibodies, and I know that I'm 55% triple knockout, and I also have my count, I've already done all my work. I know, you know, to a high level of statistical certainty how many plates I'm going to have to plate out to get that. And there's really no other way to get that data and really get it that quickly. And just the amount of time that can save, and you know, with that additional level of accuracy is just staggering. And then additionally, adding things like viability parameters as well, you know, that gives you an even better estimate of, you know, how many plates you'll need to plate out to get your desired clone. So, you know, along the same lines, but switching gears a little bit, you know, I've talked a little bit about guide RNA validation and optimization. So, you know, a lot of times we're looking at the overall, you know, knockdown, but sometimes, you know, we're trying to create, you know, nice assays to, you know, to measure our efficiency. You know, let's say if we're trying to, you know, optimize a guide RNA design or a certain type of guide RNA purification or what have you. You know, in some cases, you may not need to go through sequencing. You may not need to end up with a clonal population. But let's say you're just trying to optimize your experimental parameters and see how well it works. So here's an example with fluorescent proteins. And, you know, I just have the example here on the left because, you know, flow cytometry can definitely, you know, work very much hand in hand with cell imaging. So this is an NF-kappa B sensor cell line that has a GFP fused to NF-kappa B. And this is a nuclear translocation assay. So, you know, when stimulated with TNF alpha, right, the NF-kappa B will be in the nucleus. Otherwise, you'll see it concentrated in the cytoplasm. And I, you know, have a schematic of that, you know, that pathway there on the left. But, you know, I also used this cell line to, you know, that's stably expressing GFP to optimize my guide RNA conditions, right? And, you know, optimize some aspects of my delivery as well. And, you know, you can see that, you know, I just have my overlay with showing the GFP knockdown, and that's going to, you know, be my overall metric for my efficiency. And then I also have my, you know, my plate map down below. And, you know, now I have my percent GFP knockdown for a lot of different conditions that I tried when I was preparing different types of guide RNA. So I blinded those a little bit. The conditions are just denoted by, you know, A, B, C, and D, as well as IVT. And then I have my various controls as well, like my untransfected control and my guide RNA that's not targeting any area in the genome control. And, you know, as you can see, I have my, you know, my GFP knockdown that's normalized NF3, as well as, you know, my comparison to my genomic cleavage assay, as well as my viability measurement. And, you know, in this case, you know, I was able to analyze a 96-well plate by just really not having to do any additional processing. I just needed to lift my cells from the plate and put them in the flow cytometer and, you know, press go. And, you know, now I have all of this data that would have taken me the better part of a week and, you know, a lot of different reagents and labware and hands-on time to be able to get this by other methods. So, you know, now I'd like to show you another application. So I talked a little bit about, you know, gene tagging, and I've really focused on, you know, knockdown, but you know, another application of gene editing is being able to tag a gene. You know, once you tag a gene, there's a lot of different things you can do with that information. You know, from a gene editing standpoint, let's say you're trying to look at gene modulation, right? And so, you know, you want to create a gene tag. So we have a really nice kit. It's a TrueTag donor DNA kit for gene tagging, and you can use this for gene modulation analysis. And you know, of course, since we're tagging with a fluorescent protein, you could see that, you know, it's really valuable. And, you know, one method to, you know, analyze these types of experiments is using flow cytometry. So we had, you know, a couple different options for this gene tagging kit. We have a GFP or an RFP tag or a luciferase reporter. And you can tag either the C or N terminal of a gene, and I'll move to the next slide. And this gives you, a nice little schematic of, how this is prepared and how this workflow works. So we provide, everything needed to design this experiment appropriately. And, you can knock in the tag here, and this is using HDR, which I talked about before, because we're doing a precise modification to a gene. And additionally, in addition to adding the fluorescent marker, you can also add a resistance marker, which can allow you to then use, antibiotic selection on your cells as well. And then there are LOX-P sites, so you can cleanly, use Cre recombinase to, remove that resistance marker at the end. And this is a really nice way to tag a gene and to look at homologous recombination and look at, potential gene modulation studies. So, here's another example of the workflow and how imaging works well with flow cytometry together. And you can see that, within, day 1, you've prepared your donor DNA. Days 3 and 4, you can start to see the tag being expressed. And then at day 10, you can see that, we're 99.5% GFP positive, right, after applying selection. So, and then here's also just, some nice pictures of, looking at the, beta-actin tag using, our GFP kit. And you can see those are just really nice pictures generated from our EVOS microscope, fluorescent microscope. So, I'd just like to show you some, application data. So, umbilical cord blood is, very relevant and notoriously difficult to edit, right? And is, very important for a variety of therapeutic applications. And, here's an example of using the TrueTag, right, to be able to tag, right, this gene. And you can see that, we looked at, a few different markers, and, within 5 to 7 days, we were able to, assay these cells for using the colony forming count as well as the Attune NxT to look at the differentiation capacity of these GFP positive cells. And, note we're also able to look at the different doses and get a really nice dose response of our editing experiment as well as measured by GFP positive cells. And, these were also analyzed, to ensure that they were still CD34 positive and CD90 positive, after adding the TrueTag. And that's just, another level of rigor and, something that I touched on before and wanted to add, again, is, as you're doing these gene editing experiments, flow cytometry also gives you this, ability to look at a lot of different parameters of interest at the same time to ensure that your cells are not being affected in some other way that you did not plan on. And also ensure that you still have functional capacity. This is also, of course, very relevant for, just iPSC studies, being able to use flow cytometry to verify that your cells are, still expressing the appropriate pluripotency markers, at the end of your experiment. And, and then from a, an optimization standpoint, as I mentioned before, some of these, primary cells are very difficult to edit, even harder to edit using HDR, and, really need to go through the NEON process and need to be optimized, which is not arbitrary. So, here's an example of, using flow cytometry to get, some very nice data with statistical power showing the appropriate buffer, right, to be used for this HDR experiment using NEON. So, it can really be used through every step of the workflow from beginning to end and, save a lot of time. An additional application of flow cytometry would be, using, using flow cytometry to optimize, your viral workflow. So, if you're running a screening application or need to optimize your MOI. There are a lot of different ways that flow cytometry can come in handy here as well. So, just for, one example, I have here a schematic of our, our lentiviral library. So, one of our products is we have a library that targets the entire genome. And, with the goal to be to, assay using these libraries, knockout screens. And, one application where we use flow cytometry in developing this was to use flow cytometry not only just for our, our viral titering, but also to optimize our MOI. So, which is, varies greatly between different cell lines and, depending on your workflow and what you're trying to do. So here's an example of, HT1080 cells, infected with, viral particles that had GFP in the backbone as well. And, here's an example of using viral titering. So, as you can imagine, creating, a library targeting the entire human genome, druggable genome, about 20,000 genes. That is a lot of viral production and a lot of viral titering, and a lot of steps and a lot of things, frankly, that can go wrong along the way. So, we used flow cytometry regularly and used those GFP lenti control particles because we were producing our virus, and we used that as a sentinel as we were titering hundreds and hundreds of plates via P24, via antibiotic selection, crystal violet staining. We used the GFP titering as a sentinel that was, so much faster and, saved us, countless hours and, really money, right, in order to, check any plates, doing a QA check before we move to the next step of viral production, which is very handy. So you can really, get creative and use it in a lot of different ways. Another way I would use flow cytometry is, and this is, our standard recommendation for customers who purchase a library, is to use these lentiviral control particles to first optimize their MOI and, optimize not just their MOI, but their general viral transduction protocols for their cell line of interest. How many days they want to select, how much Cas9 virus they want to add versus how much of the guide RNA virus they want to add, and, at what time point you want to run the assay. So again, flow cytometry was a very high throughput way to, check for functionality of these different targets, right? And you can see, you're familiar at this point with the, the knockdown plots, but also look at the appropriate days of selection that, were best for this cell line to get, your maximum knockout efficiency, while also, minimizing the time that you need to keep these cells growing over time and moving forward. So, so this is, optimizing viral transduction using flow cytometry is, something that we would use all the time as well. Here's just another example of flow cytometry used in conjunction with imaging. Here's an example of, using flow cytometry to validate functional knockdown of a centromeric protein in cell. And you can see the multinucleated cells, right, versus that have been treated with guide RNA versus the Cas9 only control analyzed by flow cytometry. And then you can also see visually these cells under the microscope, and you can see, you can validate this experiment with your eyes as well. So, now that I've talked about, flow cytometry and the, advantages to using flow cytometry in gene editing, I kind of like to, just do, compare and contrast some of the methods and some of the things you can get from flow, some of the things that you can't, and, where it can really, really help and where it adds the most benefit. So, as far as hands-on time, it is, a clear winner compared to these other methods as far as how quickly you can get your functional knockout data. All of these methods require an instrument, right? There are different instruments as far as the level of maintenance that is required to do so, as well as the availability of a standard lab. So, not everybody has a 3730 or, an ion chef. Probably most labs will have, UV and be able to image a gel. So that's one thing that's handy about the GCD kit. As far as consumables cost, flow cytometry is really the clear winner as well. As I mentioned before, you don't require any additional consumables necessarily. I mean, of course, it depends on your experiment, but if you're doing, knockdown analysis, you just require an antibody. If you have a fluorescent protein-based system, you may not even require that. And again, this is, not requiring a lot of different plate transfers, a lot of pipette tips and everything else. You can just put your cell culture plate, once you've, appropriately prepared it in the flow cytometer. As far as automated data analysis, Sanger can be quite automated as well. NGS is less automated, but it still can be automated. Genomic cleavage detection, not so much. That would be, very time consuming to analyze, 896 well plates as in the example here. Turnaround time, very quick. Your answer is immediate. It is very amenable to automation as well, and some of the other methods are as well, but I would say it is the most amenable to automation. As far as environmental impact, flow cytometry is very nice because, again, it's just the reagents, the consumables to run the instrument, as well as, what other antibody you may need or what other, plates you require to analyze. You don't need a lot extra. As you can see, I have a picture here down below just highlighting the amount of waste that is required to process and, get these answers using Sanger, NGS, and GCD. Flow cytometry, there's no picture there because it doesn't require anything additional once you move that plate out of the tissue culture hood. As far as accuracy and precision, it's very high. Max throughput per day, it is definitely the highest. Sanger would be the close second, but then again, this is assuming you have a 3730 that can run 16 plates overnight. Not everyone has that. Flow cytometry will also allow you to do knock-in analysis. The others will not. It also gives you that additional metrics of cell function and viability. Now, one thing that, flow cytometry will not give you is a detailed indel spectrum. And of course, if you're creating a stable cell line, you will need to at one point, validate that cell line by other methods at some point. So, really, the take home here is, flow cytometry is not just efficient, but it's very cost effective from both, a labor and consumable standpoint. And it is very clean and safe. You're not running gels. You're not generating a ton of plastics to run these experiments, which is always nice wherever we can as scientists, right? So I'd like to just, show you one picture, of an example of the Attune. So, really, genome editing is one of the most important things is faster, right? To be able to, get our answers faster and, get these studies done more quickly, of course, with the level of rigor we need for analysis as well. And, flow cytometry really enables that. So here I have, the Attune highlighted. You can analyze up to 14 parameters. It's very small. You can even put it in a hood. It's very robust and clog resistant. From a fluidic standpoint, it is, very straightforward and does not, use a lot of fluidics. And then it's, totally automatable, right? You can, use a, orbiter, which can move the plates in and out, run plates overnight. And then we have our new Sidekick Max auto sampler, which can run plates very quickly. Before I talked about 45 minutes, well, that's now about cut in half. It's about 23 minutes to analyze a 96 well plate. We also have external fluidics and all kinds of extra automation capabilities in order to, completely automate this workflow. So in summary, flow cytometry provides a rapid and robust functional knockdown validation of edited cells. It accelerates it. It speeds it up, right? So it's truly high throughput and automated as a workflow solution. It advances cell engineering studies as well because it, allows us to do those things we talked about before as far as, gene interactions, single cell analysis of multiplexed experiments. And, again, it's, efficient, cost-effective, and environmentally friendly, which is, really a nice added benefit. And, the Attune NxT in particular with the Psychic Max is fast, right? So, it gives, it's just the perfect complement for the workflow and that it's very fast and very user-friendly. So, I'd like to just, highlight to you, I encourage you to visit our website and, learn more about some of the resources we have. We have a lot of flow cytometry learning resources online, learning centers. We have our flow cytometry capabilities guide. We have our panel builder, as well as our antibody search tools and, our molecular probes handbooks and a variety of educational webinars and, virtual entertainment that I encourage you to take a look at. So with that, I will take any questions. Thank you so much for your time and attention. And yeah, please let me know if you have any questions. And you can also feel free to follow up with me anytime using the contact information on the invite. Thank you.
Thank you, Natasha, for your presentation. We will now start the Q&A session of the webinar. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Natasha will answer as many questions as time permits. Okay, let's get started. Our first question is, what is the main value that flow cytometry adds to CRISPR experiments that other methods don't offer?
Well, really that would be the multi-parameter analysis would be the short answer. I mean, it's great advantage. I talked a lot about speed and efficiency, but the ability to get that multi-parameter analysis, looking at multiple genes, multiple functional knockdowns at the same time, would I think be the greatest benefit conferred by flow cytometry that, isn't accessible by other methods.
Great, thank you. Okay, our next question is, are there other applications or advantages to using flow cytometry in the gene editing workflow that weren't mentioned in this presentation?
Well, there's a few tools and a few things I didn't have time to get to. So, one really great advantage to using flow cytometry is we have something called the prime flow assay, which I, really recommend you, look at and get more information about. And that's an ability to actually look at your mRNA and your gene expression simultaneously at the single cell level.
Now, what is the main advantage of using the Attune NxT flow cytometer in the cell engineering workflow?
Well, the Attune is uniquely positioned, I think, because it is, so compact. It has a small footprint. You can, put it in your, hood, fits easily in your benchtop. It's very accessible and easy to use. But I think that, from a functional standpoint, it's very, very fast. It's about 10 times faster than other cytometers, and that's a huge advantage when you're trying to process dozens of plates and high throughput analysis, as you're often doing with gene editing experiments.
Thank you. It looks like we have time for one more question. Let's go with this one. Where do you think the greatest upcoming potential synergy between flow cytometry and cell engineering using CRISPR?
So that's a really good question. So I think, one thing that I'm personally really excited to see is, using flow cytometry and gene editing for gene interaction studies. So this field has never been more accessible because, with the advent of CRISPR, it really allowed us to create these, easy and, high efficiency knockouts, which has, really opened up the field of gene interaction studies. So I think, looking at these multi-gene networks and looking at these synergistic effects between genes is a burgeoning field, and then the ability, no other methodology can really analyze that like flow cytometry can, as far as looking at all the different parameters and getting that level of statistical power as you're doing it. So yeah, I think gene interaction studies is going to be, very interesting as, in the next few years, and CRISPR and flow cytometry together.
Great, thank you. I would like to once again thank Natasha for her presentation. And before we go, I'd like to thank the audience for joining us today and for their questions. As a reminder, any questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.